harvest time for wheat, the world's most important crop. Reaping that was done by hand scythes has given way to combine harvesters that relentlessly thresh every last grain. The fields producing today's record yields are full of new varieties with nostalgic names: Longbow, Fenman, Avalon, genetic newcomers that are a success story for plant breeders. The science of breeding is now being complemented by breakthroughs in the laboratory that hold the promise not only of bumper crops and high yields, but of new breeding methods for the wheat crops of tomorrow. Today's wheat plants, treated with fertilizers, pesticides and herbicides, yield harvests double or treble those of 50 years ago. Predictable and uniform, wheat now accounts for 1/5 of the world's food. The new breeds can be grown in near tropical climates, and this has meant a breakthrough in food production for countries like India, while in Britain, any surplus contributes to Europe's requirements. The vast prairies of North America are knee-deep in wheat too. Breeds here have been developed to grow in arid conditions but don't support the traditional European varieties. Thanks to these more versatile breeds, many countries are now self-sufficient in wheat, and their surpluses help to relieve the shortages in other countries. Man's selective breeding to improve crops has a long tradition. Modern wheats are a culmination of 6,000 years of domestication and breeding that began in the Middle East. Wheats arose from wild grasses that self-pollinated, although occasionally nature produced new breeds by chance. Hybrids arose from the accidental crossing of different types, and some of these had seeds which were edible and nutritious. The wild ancestors of wheat can still be found in the remote parts of the Middle East, and they're studied at modern cereal breeding stations like this one in America's Pacific Northwest. Nowadays, we can choose the best plants to improve our wheat, following the traditions of the earliest farmers who learnt from nature.
Let's keep the next one, put it in the crossing block.
Warren Kronstad is Professor of Crop Science at Oregon State University.
In the case of wheat, it's so important worldwide that I think it's easy to see that over in the Tigris-Euphrates area where wheat's thought to originate, that perhaps the first people over there saw the primitive wheat such as we see here and recognized that there were kernels inside that head and that spike, and when thrashed out, they could make a number of products for food. And as those ladies did that and thrashed that out, they realized that there was an awful lot of work to that. And as you see, there are no kernels, even after you thrash the head out. And perhaps as they did that, one day they found another kind of wheat, perhaps a mutation had happened or a change of some sort, a genetic change. And they realized that when they thrashed this wheat out, that in fact, they would get the berries flashing free. And as a consequence then, as these wheat seed you see here, it made life much easier for them. And then they recognized if they took those kernels and planted those back the following year, that they would get wheat like this, which the grain would thrash free, rather than wheat like these primitive types.
Selective breeding became more organized to meet the growing demands of an increasing world population. Over the past 100 years, new wheat varieties have been grown and exported on a massive scale. European settlers in North America developed new varieties of wheat that would ripen in the short season of the prairies. For Canada, output increased fourfold in just 5 years after the new crops were introduced. In Australia, still other varieties were developed which would tolerate very dry conditions. They would also ripen in the short days of the near equator. By 1930, Australia rivaled northern European countries as a major wheat producer. But it's the last 40 years that have seen really major changes. Until then, varieties were tall. The ratio of grain to straw was low, and if fertilizers were added to improve the yield, the stalks couldn't support the weight of the grain. The wheat simply fell over. Crossing these tall plants with dwarf varieties from Japan gave us our modern semi-dwarf types with shorter, stiffer stems, but the same amount of grain. A classical crossing game and a predicted outcome. But to mature and ripen, the wheat plant needs summer days of 14 hours, so equatorial regions couldn't grow wheat. Further crossing with varieties from Australia solved this problem too, and modern wheat no longer requires prolonged bouts of sunshine.
As a consequence, the Green Revolution was launched, and countries like India and Pakistan and Bangladesh were the beneficiaries of the so-called Green Revolution. And that must represent, I think, the most classic example in wheat breeding of a tremendous success story and how plant breeders have modified the wheat plant, made it more widely adapted to meet the needs of people.
Varieties incorporating dwarfing genes are now sown the world over, and they've made a great impact on countries like India and Bangladesh. Varieties developed at the International Wheat and Maize Improvement Center in Mexico transformed agriculture in parts of the Indian subcontinent in the 1960s. Yields per hectare doubled overnight. Wheat breeders see this as the latest in a long line of achievements for their science. Inspired by these successes, Britain's Plant Breeding Institute continues its search for new breeds. Although there are now over 50,000 known wheat varieties, only a handful are judged successful enough to be used on a commercial scale. So how do you make a better breed? Before crossing one variety with another, breeders have to prevent the normal self-pollination by emasculating the immature plant, physically removing the male anthers that normally produce the pollen. A different plant will provide the pollen to fertilize the female stigma. Each fertilized stigma will subsequently form a seed which will produce a hybrid plant. This is the basic procedure for crossing, and up to 1,000 different crosses are made in attempts to combine the genes for the new variety. It takes about 3 weeks just to make all the crosses, and it's done every January. By March, seed from the crosses, known as F1 seed for the first generation, has been planted out in cold cabinets. Most wheat is winter wheat. It's normally planted in the autumn and has to experience winter before it will mature to give seed. The cold room is simply a means of speeding up the process. The F1 plants will mature and self-pollinate to give F2 seed in time for autumn sowing. The F2 seeds are now packaged in individual envelopes, so each envelope represents a separate cross, and each cross is sown as a separate row. The following spring, as each F2 plant within a row is genetically different, will the differences become apparent? It's the first opportunity to sort the good from the bad. Former head of cereals at PBI, Francis Lupton, explains.
Well, we're in the middle here of the F2 generation. We have here the plants from the crosses which we made last year, and each cross gives us a row like this of about 2,000 plants. And you can see these rows vary very considerably in height. We have here a tall plant, here we have a nice short one, more short ones in behind here. We also have material which is varying in disease reaction. For example, this plant has too much yellow rust and would have to be discarded. Whereas if we look at this one down here, it has far too much powdery mildew, and that's no use for a future variety. And what we do is to go through this material at this time of year and mark with colored tags the plants which look desirable. A plant like that, or like that one, or perhaps like this one up here. Then we come back again in 6 weeks' time at harvest. If it still looks good, we harvest that plant, and that provides us the seed for the next generation.
The original 1,000 crosses have produced a field full of F2 rows. Every plant is genetically different. Amidst these varieties is the one that's wanted. Why must so many plants be grown to obtain any one variety? It's a question of selecting from the many possible combinations of genes produced in the crossing. Let's imagine the case for just 3 gene pairs, A, B and C from each parent. Capital A, B and C give short, pale awns, small a, b and c the opposite. Say we're aiming for a short, dark, awnless plant, big A and small b and c. The F1 generation is intermediate, a pooling of both versions of each gene. Our plant, big A, small b and c, will be in the next generation, along with many other types. Laid out in a grid, we can see all the possible combinations for the F2 generation, the result of pooling 2 sets of 3 genes. There are 64 gene combinations for the offspring, and only one of these is the one we want. We've only looked at 3 genes. If we select for more genes, the number of F2 plants we need to grow soon becomes astronomical. So, breeders have to weed out a large number of plants to leave just the ones they want. It takes several generations after the original cross before they can be confident that all the unwanted gene combinations are eliminated. The promising varieties are grown in trial plots. It's 6 years since the first cross that produced these plants, but the breeding isn't finished yet, as the genetic uniformity of each variety can only be established over several more generations. Take the case of a gene associated with height. Say we have 2 lines which differ for this gene, one producing tall plants and the other short plants. Let's run through the generations. In the F1 generation, all offspring are intermediate in height because they contain both forms of the gene. In the F2 generation, only half of the plants will be intermediate. The others will be uniformly tall or short. At each subsequent generation, the proportion of intermediates is halved. After 10 generations, intermediate offspring effectively disappear from the population, leaving uniformly tall and short plants in equal proportions. Now, we've only looked at one gene, but breeders attempt to combine several characteristics. So achieving this uniformity can take up to 10 years after the original cross. Traditional plant breeding is a time-consuming business. Nowadays, we have the exciting possibility of reducing the multiple crosses to one simple step. Genetic engineering, injecting DNA into single cells, offers the potential to introduce only the genes that are wanted from the outset. But for plants to be grown artificially, a special surgery is also being invented. Inside this seed is a growing embryo. By clinically removing it and culturing it under controlled conditions, the important stages of development can be more easily identified for gene transfer experiments. But tissue culture in this scientific greenhouse has yet to produce a fully fledged cereal plant. The problem has prompted researchers at the Max Planck Institute in Germany to try a much simpler approach for transferring genes into cereals. Their idea was to introduce the DNA of a single gene into fully mature plants even before seeds are formed. Head of the team is Horst Lertz.
Our interest was to look for a possibility for gene transfer, avoiding tissue culture, and if possible, transfer the DNA directly into a plant. And of course, if possible, the best way would be if we could transfer the DNA directly to the cells which finally form the flower, or let's say the so-called germ cells, which finally form the pollen grains or the egg cell.
They've identified the critical stage for injecting the DNA into rye plants. It's a few weeks before the pollen grains develop. As the shoots appear in a batch of plants, one at just the right stage of development is selected, and the DNA is injected. But can they check that the DNA is taken up? To do this, they use the DNA of a marker gene that makes plants resistant to the drug kanamycin. So, if the experiment works, the plant will acquire a new resistance to the drug. A few months later, the seeds from the injected plants are growing on a medium containing kanamycin. Most of the seedlings are white. The drug is inhibiting the production of their green chlorophyll pigment. But a small number of them produce green plants, a clue that they've become resistant to the drug and that the injection has worked. For definite proof that the gene has been passed on to the new generation of seedlings, the research team took up the traditional methods of DNA analysis. By isolating the plant DNA and analysing its characteristic pattern on fluorescent gels, they confirmed that the injected DNA was not only present, but had retained its activity in the plant. Their method has the potential for introducing the genes for disease and pesticide resistance, and for improving grain quality in other cereals. So, will genetic engineering affect traditional plant breeding?
It for sure will not replace breeding activities. The biotechnology or the new methods in genetic manipulation for sure might help to speed up a breeding progress or make it more efficient, but they will not replace it. So what the real advantage I see is that you can do plant breeding much more defined. So far, when you combine two genomes, you had hundreds or even thousands of genes combined, and the major effort in breeding, if you want to make it short, was to cross out most of the undesired traits. But now you can transfer just one single character by using one defined gene, and that really will help you to make the result much more predictable, and that I see as a real advantage of genetic manipulation procedures.
The genetic engineering of cereal plants promises exciting new breeds, but it will never be totally divorced from experiments in the field. Developing new crops will continue to depend on a field approach that rests on a breeder's experience. Warren Kronstad explains.
We have to be very honest, I think, as plant breeders and say that plant breeding is both an art and a science. Certainly the science is the genetics and being able to predict how various traits are going to come out in genetic ratios. But there is also an art to it, and that comes with experience. I doubt if there is a plant breeder in the world who could honestly define and describe the ideal wheat plant. But we do know that there are certain parental types which we say have good combining ability, that when crossed, they tend to give us a high percentage of good progeny with good disease resistance and good winter hardiness and all these other factors we must have in our varieties. Oh, that's beautiful seed. Yeah, that's what we're looking for. Now let's take that one right to the yield trial. Okay. And then put it in a hard red winter crossing block.
Improving wheat ultimately comes down to concentrating on just one or two successful varieties. But there's an inherent danger in relying on such a limited gene pool for wheat. Norman Simmons of Edinburgh University was one of the first people to recognise this problem over 30 years ago.
It's a reflection, rather, of the narrow genetic base of breeding in the past. Highly effective plant breeding always limits the numbers of materials that actually get into agricultural practice. The better they are, the fewer they are. Now, there are some possible risks along the lines of very small numbers of varieties, maybe being susceptible to unforeseen diseases. And there are certainly instances of this having happened in agriculture and in a good many places in the past. And no doubt they'll happen again in the future. So there are some risks then in a narrow genetic base, but few varieties are an inevitable feature of a high level of adaptation and performance. You can't have huge numbers of excellent varieties. It just isn't possible.
So breeders continue to focus their efforts on just a handful of crop lines. This has been successful in meeting today's needs, but what of the future?
In principle, perhaps one could reduce all our crop plants to just 2 or 3 lines. But that guarantees non-response to the needs of the future, to changed requirements on the part of farmers and consumers, to new diseases, to new markets. One must have the materials for response to future needs. And since we cannot, by any means wholly, or even if we could foresee what those needs were, we would still need a vast array of genetic material in order to be able to search through it, to find the stuff that's wanted against the requirements. To be able to think of replacing genetic variability by waiting for mutation or making mutation has been suggested, but in my opinion, it's just plain silly. It's far cheaper and more efficient to keep the stuff in the first place.
This collection of seeds from different varieties is being stored for the future. Each of these packets contains a separate seed variety. This gene bank is a natural resource, worth conserving, so that, if necessary, chosen genes can be reintroduced into a breeding program. Unfortunately, in the few gene banks that exist for wheat, the primitive wild varieties, which have evolved wide disease resistance and are therefore most valuable, are scarce. The computer is an ideal tool for storing all of the field data from breeding programs around the world. As the demand increases for intricate crosses and complex varieties, it's likely that the computer will play a larger role in the design of breeding programs for the future. It will help in the search for resistance to numerous diseases, the best proteins for bread, and of course the best yield.
Profile. That's really an international cross.
The new breeds of wheat have helped revolutionize agriculture and provide a basic food for the nations of the world. We have all benefited from the success of applying genetics to improve crops over the last 30 years. As the proving ground for plant geneticists now includes the laboratory, the future holds even better prospects for the new breeds of wheat.